lipids and lipid digestion. Specifically, we followed the digestion of triglycerides from the mouth all the way through to the small intestine, where that last step happens. The fatty acids and glyceride come in. The intestinal epithelial cell puts that triglyceride back together, packages it as a chylomicron, right? So in the end, the lipids that are ingested end up in the lymph, not in the blood, but in the lymph, in these packages of protein surrounding a lipid core that we call a chylomicron. All right, so the journey isn't done there, because from there, the chylomicrons now have a journey all their own. So we pick up here in this box. So the lipid is now packaged inside a protein shell, and that protein shell with its lipid inside is dumped into the bloodstream. So the chylomicrons enter the lymph in the small intestine. That lymph travels up the thoracic duct, which is the large lymphatic vessel near the aorta, and eventually it arrives at the left subclavian vein. So that lymph gets dumped into there, the chylomicrons get dumped into there. So we go thoracic duct, and then eventually we enter into the blood. So from there, so these chylomicrons are chock full of lipid. So the organs and places that can use that lipid, they're going to sort of make a withdrawal from those chylomicrons that are circulating. So along the way, from subclavian vein to liver, some capillary walls have an enzyme called lipoprotein lipase, which essentially breaks those chylomicrons and causes the lipids that they contain to be released into those tissues. So some example tissues where that's the case are skeletal muscle up here and adipose tissue. So adipose tissue stores lipids, right? We all know that. Fat tissue. So if we have an excess of fat coming in through the GI tract, the adipocytes or adipocytes, they can take that fat directly out of the chylomicron and store it inside their own cells. So some of the lipid that's been ingested ends up directly adding to the adipocytes that are already present. And then skeletal muscle actually prefers fatty acids as its fuel source because they're so energy dense, as we'll see here in a minute. You can get more ATP out of a fatty acid than you can out of a glucose molecule. So another place that those lipids that are absorbed end up is inside skeletal muscle, where they're used to create ATP and to sort of power skeletal muscle tone, which is the small amount of contraction that's present all the time, as well as generate the ATP to turn glucose into glycogen. So we have adipocytes and skeletal muscle. But the sort of core of lipid metabolism in the body is our friend the liver again. So just like the liver manages carbohydrate metabolism, it also manages lipid metabolism as well. So the chylomicrons that are not broken down and used by the skeletal muscle or the fat cells end up at the liver. So here we are right here. Chylomicrons enter in. Now, once they're in the liver changes them. So the triglycerides that are present in the chylomicrons, those are removed, and then the protein package is re-released. But because the triglycerides have been removed, we don't call them chylomicrons anymore. Instead, we call them LDLs, low-density lipoproteins. So that's what the LDL. What does that mean? It just means that it's a chylomicron without the triglycerides. All right? So LDLs are proteins that carry lipids, particularly cholesterol, which is another lipid, out to the tissues where they can be used. So once the LDLs get out to the peripheral tissues, they're absorbed by cells that need them. The cholesterol is released, And then that cholesterol becomes part of the cell. You know, we hear a lot of bad things about cholesterol, but it is a required nutrient. You know, like because cholesterol plays a role in cell membrane structure, we have to have a little. So the cholesterol from the food that we eat ends up traveling to cells via these packages called LDLs. All right, so the cholesterol is released. Well, what if there's too much cholesterol? How do cells get rid of it? Well, they get rid of it by packaging it into HDLs, high-density lipoproteins. Those HDLs are returned to the liver. Their cholesterol is released in the liver, and then the excess cholesterol is dumped into the bile and into the small intestine, where then it's removed with the feces. Because the kidney, it can't get rid of lipids very well at all because of how the kidney functions. So the only way we can get rid of excess lipid is through the bile. So you may have heard of these two lipoproteins before, LDLs and HDLs. That's the good cholesterol and the bad cholesterol, right? And we can see why they get their names. So LDL is the bad cholesterol. And the reason for that is LDLs are carrying cholesterol out to the cells of the body, which is not where we want it. You know, if we have a person who has atherosclerosis, let's say, cholesterol plaques in their arteries, the last thing we want circulating around is cholesterol delivery proteins, right? So LDLs are the bad cholesterol because they're carrying cholesterol out to the body to be used. HDL is considered the good cholesterol because HDL is carrying cholesterol in the opposite direction. A high HDL level means that your body is getting rid of cholesterol, which is exactly what you want to see in a patient who has atherosclerosis or vessel disease. You want to see them dumping cholesterol, not distributing it. So LDLs and HDLs you're going to hear about in your careers and on exam 2 because of their clinical relevance. You know, you can now understand why is LDL bad and HDL good. It has to do with the direction the cholesterol is moving in. You know, LDLs it's going out, HDLs it's going back to the liver to be removed. And all three of these, chylomicrons, LDLs, and HDLs, are all part of a class of proteins we call lipoproteins, which means they're part lipid and they're part protein. All right? So LDLs and lipid transport. All right. So here's another one of those messy chemistry-looking slides. But rest assured, this is not a chemistry class. So I'm going to reduce what I think you need to know. All right. So beta oxidation is the way lipids are broken down. You know, there's no point in storing lipids as triglycerides, like in adipocytes, unless we can use that energy somehow. So lipid molecules are very, very large, usually much larger than the simple amino acids and carbohydrates that we talked about. So like in this example, this molecule is a fatty acid, and it has 18 carbons. So what do we want to know about a fatty acid? One is, if you look at this structure, you see very little oxygen, right? In a carbohydrate, there's an oxygen for every carbon molecule. In a fatty acid, there's only 2 oxygens in the entire molecule, even though it's got a whole bunch of carbons. And the oxygens are here at the end. So what these molecules look like look like this. They look like long chains of carbons, and then they have sort of an active end that can be attached to something or removed from something. All right, so that's a fatty acid. Way, way too big for the mitochondria to break down all at once, right? Last time we talked about the mitochondria can only handle 2 or 3 carbon chains, right? So little tiny pieces. So in order to break this molecule down and use it as fuel to make ATP, we're going to have to split it up. And essentially, what beta oxidation is we take a fatty acid apart 2 carbons at a time, okay? So, we've got this long chain, and we're going to break the first 2 carbons off, and then we're going to break the next 2 carbons off, and next one, and so on and so on, until we don't have a fatty acid anymore, until all of the carbons have been broken off. So what we store is triglyceride. So here's 1, 2, 3 fatty acids, and then a glycerol backbone, right, like we talked about last time. So the first thing we have to do is take the fatty acids off. So we break it here, here, and here, and we have now fatty acids and a glycerol backbone. So the glycerol, it gets fed into the mitochondria after being broken down. And then the fatty acid, we're going to break 2 carbons off at a time. So here, if we look over here, so here's a carbon, and we're going to break it. And then those 2 carbons are going to go into the mitochondria to produce ATP, right? Just like we did with glucose. First we broke it, then we fed the parts into the mitochondria to make ATP through that Krebs cycle or citric acid cycle and the electron transport system. So we do this 2 carbons at a time. So now we have a 16 carbon fatty acid. You see we went from 18 to 16 because the 2 went into the mitochondria, and now this whole process can happen again. So we'll go from 16 to 14, 14 to 12, 12 to 10, and so on. So 2 carbons at a time. So by doing that, by feeding the mitochondria 2 carbons at a time, we can make a huge amount of glucose, or I'm sorry, a huge amount of ATP. So we learned that for a glucose molecule, in the end, all told, you get 36 ATP, right, for those 6 carbons. Well, fatty acids having hydrogens instead of oxygens attached to their carbons actually contain more energy. So for 6 carbons from a fatty acid, we'll get 51 ATP. So significantly more energy per molecule or than we see with glucose. So beta oxidation, 2 carbons at a time, feed to the mitochondria, 51 for every 6 carbons. So higher energy state. All right. So see, that wasn't so bad. Not a ton of chemistry there. Well, okay, we break down lipids, but we also want to be able to make them. And the chemistry there is equivalently complex. But essentially, lipids are made in the reverse of beta oxidation. So in beta oxidation, we clip 2 carbons off at a time. In lipogenesis, we add 2 carbons at a time. So essentially, we sort of reverse that process, and our chain of fatty acids, or our chain of carbons, gets longer and longer until it reaches the length that we need it to be. So that happens right here. These acetyl-CoAs, which are 2 carbon fragments, are sort of added together 2 at a time to make fatty acids. Fatty acids can be added to a glycerol backbone to make triglycerides. And then fatty acids can be modified to make all different kinds of lipids. You know, we talk about triglycerides, but lipids are a whole class of nutrients. You know, there's cholesterol, there's steroid, there's short chain, long chain, medium chain. There's all kinds of different fatty acids. And once the fatty acid is made, it can be changed into those various states. It is important to note that, or let me back up. One of the things that cholesterol can be changed into is the steroid hormones. So these are things like aldosterone, estrogen, progesterone, testosterone. Those molecules actually begin as cholesterol and are then modified to become those steroid hormones that we talked about in our endocrine chapter. Remember the hormones that could pass right through cell membranes to the nucleus? Well, those molecules begin as cholesterol and are then modified. Okay, so that's one point. Another is there are some, because of how we build lipids in our bodies, there are some kinds of fatty acids we can't build. So, for example, we can't insert a double bond into our fatty acids. So, like, all of the ones we make look like this. They're called saturated fats or saturated lipids, where there are no double bonds. But there are a few things in the body that we need a double bond to make. So all that to say, we have a couple of essential fatty acids. In other words, lipids, fatty acids, that we have to take in our diet because we can't make them. So the 2 that you'll hear about are linolenic and linoleic acid. So if you see LINO, that's one of the essential fatty acids, okay? So those are the omega-3s and omega-6 fatty acids that you'll sometimes hear about in nutrition. So all I want you to know is there's two essential fatty acids, and they both start with lino. So linolenic and linoleic acids. And we need those so we can get that double bond that we can't make. And then we can modify it into what we need. All right. Okay, I think that's everything on this. Okay. All right, so from lipids to proteins. So we talked last time about carbohydrates and lipids. Today we just finished lipids and lipid metabolism, breaking them down and making them from scratch. So the last one is proteins. So if we follow proteins from ingestion to metabolism, it looks a little bit like this. So protein in the food that we eat is chewed up, mixed with saliva. There's no protein digestion that occurs in the mouth. There is fat and carbohydrate digestion, like we talked about, but no protein digestion. So we chew and mix and swallow, and then when we get to the stomach, the acid in the stomach works to denature the protein in the food that we've eaten. And what that means basically is to unwind them or uncoil them. Proteins have a tendency to be all twisted up, like a wire that's been twisted and twisted and twisted. So denaturing uncoils that wire. And then pepsin, which is an enzyme the stomach makes, starts breaking those long chains of amino acids into smaller pieces. So we get smaller polypeptide chains. So that's there, number 2 in the stomach. And then third, the duodenum. So the stomach empties its contents into the duodenum. And the stomach can't finish protein digestion. It can only start it. Because pepsin, it can't attack proteins that are very small. It can only attack proteins that are sort of large. So we're not done yet. So the pancreas adds its enzymes called proteases. A protease breaks down proteins. And the pancreas has sort of a toolbox of enzymes that attack chains of amino acids. So its enzymes can turn proteins into very small pieces, right? So that occurs in the small intestine. And then finally, what we end up with are a peptide is another name for an amino acid. So after the pancreas is done with it, we end up with very short chains. So 3 and 2 amino acids stuck together. So like this is called a dipeptide, right? And this would be a tripeptide. So by the time the pancreas enzymes and pepsin is finished, we've turned this long string-like thing into a bunch of eventually dipeptides, little tiny bits. And just like we saw with carbohydrates and lipids, the last step, which is to make amino acids now, so break down it into its component part, that last step happens right at the brush border. So as these very short peptides are being brought into the intestinal epithelial cell, those last few bonds are broken. So what are absorbed from protein that we eat are individual amino acids, right? And then those amino acids get absorbed into the bloodstream, distributed, and then we use those amino acids to now build our own proteins. So from the protein we eat, it's broken down, eventually to its component parts, absorbed, and then put back together again into whatever it is that we want to make, that our cells want to make out of it. So last step occurs at the brush border, just like the other 2 nutrient types. All right. So there are a wide variety of amino acids, and in order for our proteins to function correctly and be shaped correctly, we have to have exactly the amino acid we need in exactly the right spot, right? We've learned that last semester in other biology classes. Well, that means that, but the diet, our diet varies, and not all the things we eat have all the amino acids we need. So we need to have a way to make amino acids that we're short on. And the liver does this. The kidney does a little bit of it too. But there are multiple ways that the liver can produce the amino acids that we need. So two such ways are these two. So amino acid synthesis. In other words, we're going to create an amino acid. One is a process called amination. And that's where we can take a molecule that's a carbohydrate. Do you see how there's no nitrogens in this molecule, right? It's all oxygens and carbons. Well, we can take a keto acid, this is called, or a carbohydrate, and we can add a nitrogen to it, because remember, amino acids are different than carbohydrates because they have a nitrogen molecule that's part of them. Well, we can add that nitrogen by adding a molecule of NH4. NH4 is ammonia, right? Same stuff that you would use to, clean the kitchen floor or the bathroom floor. So we can add an amino, ammonia. You can also call that an amine. We can add that ammonia, and what do we get? We get now this is an amino acid. So we've essentially made an amino acid by adding an ammonia molecule, and we get a water out. So that's one way the liver can make an amino acid. Another is by taking an amino acid and a carbohydrate and swapping the nitrogen, because it's the nitrogen that's going to make that bond work. So we call that transamination, where we take the amine group from one amino acid and we flip it to another. So like, let's say the body has too much glutamic acid and not enough tyrosine. Well, it can do this little trick, and it can produce tyrosine from excess glutamic acid. Okay? So what do I want you to know here? Amination is adding an ammonia to make an amino acid. Transamination is switching the amine from one molecule to another to sort of swap and make a new amino acid. Now, there are 10 amino acids that we cannot produce. So we call those essential amino acids. And that they have to be acquired in the diet because we can't produce them. Of course, I'm not going to make you know what those 10 are. And for the adult, there's only 8. But we say that there's 10 essential amino acids because for children, those other 2, they can't produce them fast enough to match their growth rate. So in general, people will talk about there are 10 essential amino acids. All right. Well, what about breaking amino acids down? What if we have a high protein diet, like a carnivore, like a house cat? Most of their diet is protein, right? So once you've made all the proteins you're going to make out of amino acids, well, you'd like to be able to use the excess as fuel. So in order to catabolize or break down these amino acids, we have to get rid of the nitrogen atom that is part of them. The mitochondria can do nothing with a nitrogen-containing compound. So in order for the mitochondria to do its stuff, we have to pull the nitrogen off first. And that process is called deamination. So it's the reverse of amination. Amination makes an amino acid. Deamination turns an amino acid into a simple organic acid or a carbohydrate. So in that process, we form an ammonium ion. And this is a problem because ammonium, as you might imagine, is very, very toxic. You'd never drink that stuff you use to clean the bathroom, right? And for good reason. So if ammonium is being produced, the body's going to have to do something with it very quickly. Otherwise, it's going to build up and that cell is going to die. So what it does is it takes the ammonium, it adds it to a carbon dioxide, what the cells do. They take an ammonium, add it to a carbon dioxide molecule through this process called the urea cycle. And what you get out is this compound that looks like this. So it has 2 nitrogens, an oxygen, a carbon, and a bunch of hydrogens, right? And urea is relatively harmless even in large amounts. So this ammonia is transformed into something harmless. Then the urea is expelled into the bloodstream, and eventually urea is filtered at the kidney to make urine. That's how it gets its name. Urea is the nitrogen stuff in urine. So you get the urea cycle, and it's the whole purpose is to get rid of the nitrogen from the excess amino acids, so then we can feed these into the mitochondria and make energy from them. So one of the reasons why carnivores' urine often has a very pungent ammonia-like smell to it is because their diet is almost all amino acid. So they have a ton of nitrogen that they're getting rid of all the time. So they not only have urea, but they have some ammonia systems as well to get rid of that excess. All right, so amino acid metabolism, deamination, urea cycle, onward. Okay. All right, let's do a question or two. So in the process of cellular respiration, that's glycolysis plus what takes place in the mitochondria, each molecule of glucose yields how many molecules of ATP? All right, 10 seconds, last person. All right, so the best answer there is E, 36. Glycolysis yields 2 ATP, but that 2 can be critical sometimes because glycolysis can take place without oxygen being present. So think anaerobic muscle metabolism. But the oxidative phosphorylation part, that's the Krebs cycle, or the TCA cycle, and the electron transport system together, we call that oxidative phosphorylation. That makes 34. So you take 2 plus 34 and you get that 36 that I said I wanted to know. All right. Sometimes called good cholesterol. What carries excess cholesterol back to the liver? All right, there we go. That everybody answered. All right, so good cholesterol is the HDL. So do be able to tell us apart. HDLs are carrying cholesterol back to the liver, then the liver is going to dump it. It's going to dump it into the GI tract through the bile, and that's how we get rid of excess lipid. The LDLs are the bad ones because that's cholesterol being delivered to the cells of the body, which is the last thing you want if you've got a cholesterol issue. And the new book doesn't cover these other lipoproteins, so you can safely ignore those. So it would have been in between HDL and LDL. All right, the conversion of ammonia into a less toxic substance produces which of those? All right, good. That is C, urea. Ammonia is what is created when you turn an amino acid into something the mitochondria can burn, right, like your carbohydrate or an organic acid. But that process creates ammonia. Ammonia is very harmful, toxic. So the body takes the ammonia, attaches it to a carbon dioxide molecule, and forms urea out of it. instead, which is much, much less toxic. All right. Okay, so in the body, there's essentially two metabolic patterns that we encounter, in regular normal life, and that's the absorptive state and the post-absorptive state. So, we eat. We eat 3 times a day, or even more often than that. And anytime that we put nutrients into the digestive tract, eventually those nutrients are going to be absorbed by the digestive tract and are going to be available for the body to use. So the amino acids, the simple sugars, and the fatty acids that are digested and absorbed, can be used by the cells that are in need. So when the body is in that state, in other words, when there's stuff coming in through the GI tract, we call that the absorptive state. And it's and when that is happening, the body is basically gathering up nutrients. So it's going to build up, it's going to store, it's going to utilize those nutrients that are coming in. But there are times when we don't eat, like we go to sleep at night and we don't eat for 8 hours. Well, in that first thing in the morning, we still need nutrients to keep the brain alive, keep the cells functioning. So how is that possible if there's nothing coming in through the GI tract? That is what we call the post-absorptive state. And in that state, the body is sort of set to use the nutrients that it has stored for the purposes of maintaining the glucose level, keeping the cells functioning when there isn't nutrients coming in from the outside. And you could say there's a third metabolic pattern which we don't talk about, and that's the starvation state. And that is where you've been in the post-absorptive state for so long that your nutrient reserves are used up. And then the body does some other things, but we don't talk about that one, just so you know there is a third one. All right, so in the absorptive state, what does that look like at the cellular level? Well, in the blood, glucose levels are elevated. Insulin is being distributed because in the absorptive state, glucose is coming in. It doesn't have to be made. So all the cells might as well use it because it's readily available. So insulin is released. That triggers the cell to bring glucose in. It also triggers the cell to make glucose its primary fuel source. So the red arrows are catabolism, and the blue arrows are anabolism. So when insulin is present, it means that glycogen is broken down into glucose. Glucose is fed into the mitochondria to create ATP. So if that's fueling the cell, it has plenty to build up the other two nutrient types. So amino acids are made into proteins, fatty acids are made into triglycerides. So when glucose is being used, the cell is going to use these other nutrients to build and grow and develop, right? So we have the absorptive state. In the post-absorptive state, you can see the arrows in the opposite direction. So when there's insulin around, glucose is running the show. When there isn't insulin around, other things are being fed into the mitochondria. So like you can see that principally it's fatty acids and proteins are being used because glucose is now in short supply. It's not coming in through the GI tract. The cells, the neurons of the brain require glucose to be present. So in the absence of insulin, do you see how there's no insulin marker up here? The cells are sort of told, don't use glucose. Spare the glucose for the brain and use other fuels instead. So we have fatty acids being used and amino acids being used instead. Now, what this represents here is what we call gluconeogenesis. The liver, in particular, can create glucose when glucose is not coming in and not being stored. So, say you're going to go to sleep at night, and about 4 hours into that, you're done absorbing glucose from your last meal, right? Well, the liver can provide for the brain for about 4 hours from its glycogen stores. Remember, glucose can be converted to glycogen, long chains of glucose. So the first thing the liver does is it breaks down its glycogen releases the glucose so that it can be used by the brain. But eventually that glycogen runs out. And when it runs out, then the liver shifts to this, where it starts producing glucose from other things. So the liver can make glucose from various amino acids, from a substance called pyruvate, which is a small carbon fragment. It can even transform the carbons from fatty acids into glucose as well. So when the storehouse is empty, so to speak, the liver starts producing glucose in other ways. All right. So in addition to regulation of glucose, the liver also can produce amino acids, like we talked about a minute ago. So in that post-absorptive state, the liver may be creating amino acids to release as well, and it can also release fatty acids also. So the liver very much controls what nutrients the body is using at any given time, and it can produce all of those nutrients as well. So it's an organ that's focused on nutrients for the body. All right. So post-absorptive state, spare the glucose for the brain, use everything else, and then the liver can produce what's needed. All right. All right. So we've all heard of vitamins, but maybe you haven't heard of what specifically, what does that word mean? And I have to be honest with you, I never learned the vitamins and what they do, so I'm not going to make you learn them either. And in my career, it never came up, except on board exams. So I'm going to give you a sort of a real brief version of what I want you to know of all these tables with all of this stuff in it. I want you to know there are two classes of vitamins. There are fat-soluble vitamins and water-soluble vitamins. There are only 4 fat-soluble vitamins, and they are A, D, E, and K. So you should know that. That is a little tidbit of information that does appear on board exams and things like that. Now, what makes fat-soluble vitamins different? These are the vitamins that you can become toxic from. In other words, you can have an overdose of A, D, E, and K. And the reason for that is because these vitamins are stored in lipid, not in water, the body can only get rid of them so fast, right? So A, D, E, and K are ones you have to be careful with in terms of supplementation because you can overdose on A, D, E, and K. So you see that there's effects of deficiency and effects of excess, and it's because you can excess these 4. Okay? Let's see. And D3 and K. D3 is a calcium hormone. It's why it's added to milk, right? Vitamin D is added to milk. Without vitamin D3, we can't absorb calcium from the food that we eat. And then vitamin K, we've talked about already, I think. Vitamin K is produced by bacteria in the large intestine, and they're critical for the clotting factors. All right? So those are 2 little bits. And then these are the water-soluble vitamins. Again, no need to memorize all this stuff. I'd rather have you spending time learning other things than these vitamin things. So these are the B vitamins principally. There's the C vitamins. The water-soluble vitamins are not stored, which means that they have to be a regular part of your diet or you can very quickly get into a vitamin deficiency, right? So the only 2 that are stored in any significant quantity are B12 and C, all right? So all the others you have to have as a regular part of your diet. Now, in this country, we don't see vitamin deficiency, not even in kids or the elderly. And the reason for that is the government in the 1920s and 30s started the system of vitamin fortification. So like when you go to buy flour here in America, you'll see that it'll say fortified. What that means is that vitamins and iron usually have been added to it. And the reason that that's done is it was a government program because there is like wheat flour in all kinds of things that we eat, right? So how do you stop vitamin deficiencies? Well, if you can put the vitamins in something that everybody consumes, you can end the vitamin deficiency. So that's exactly what they did. So many of these B vitamins are added to the flour that becomes the bread or becomes the cereal that we eat. Yeah.
So is that what they do to all those kids' cereals that they just add all these vitamins to
Yes.
make people think that they're healthier or something.
Well, they're healthier than they are without it, yeah. And, yeah, so like cereal is a
They
classic
always
one.
talk about how they're fortified with all these vitamins. And it's like, well,
Yes, they have taken and they've added these to the flour that makes the cereal. Yeah, yep. Which isn't a bad thing. At least we don't see vitamin deficiencies.
That's true.
All right, and then of course your book has the obligatory, 2-page spread on nutrition, which we've all heard about before. So we're not going to talk about it. All right. So the last piece of this chapter is what we call energetics and thermoregulation. So energetics is our body's use of energy. So we've seen how energy is produced, right? The mitochondria produce huge amounts of ATP. Now, in the process, the mitochondria also produce heat. So mitochondria do heat up. And, of course, that heat has to be gotten rid of or else the cell will die. It'll burn itself up. So the basal metabolic rate is the energy you need just to stay alive, right? So this is, you haven't done anything yet. This is for an awake, alert person, sitting at rest, doing nothing. So doing no motion, doing nothing more than keeping themselves alive. For most people, that is about 1,680 calories per day. Now this can vary. Some people have a higher basal metabolic rate, some people have a lower one. But the average, so to speak, is about 1,600 calories per day. So that's just to stay alive, be in homeostasis. You haven't done anything yet. So if we then add activity or energy expenditure on top of that, we can see how the calories can go way, way up. So like slow walking, like a normal paced walking, that burns about 200 calories per hour. And then you can see that like competitive swimming and rowing can be upwards of 1,000 calories per hour because of all of the different muscles that are being used and the resistance to those motions. So big changes in how much calories we use. All right. So ultimately, in order to balance our caloric intake with our energy expenditure, there has to be some system to help the conscious mind determine how much food should be consumed, right? So there has to be a system for making us hungry and for making us not hungry. And both of those systems, the hungry system and the satiety, is what not being hungry is called, technically speaking. Satiety is not hungry, not interested in food. Both of those come from the hypothalamus. So there's a feeding center that stimulates hunger and hungry behaviors. You looking in the kitchen, opening cupboard doors, trying to see what's in there. Those are stimulated by the hypothalamus feeding center. You're looking for food because you're hungry. And then the satiety center is triggered in response to food being in the GI tract. So when there's a sufficient quantity of food in the stomach or in the duodenum, it sends a signal to the hypothalamus to make you not hungry anymore. So why don't you just keep eating and eating and eating and eating? Because eventually your GI tract tells your hypothalamus, that's it, we're done eating, we're not hungry anymore. So we have these two things that are in balance. And with the obesity epidemic in the country, of course, huge amounts of research are being done on just how these two things work. Because I'll get to you in just a minute. Because one path to assisting people with weight loss would be to affect this feeding center or this satiety center. Either turn the feeding center down or off, or turn the satiety center up. Yes?
So when people have a drop in blood sugar, but they're not hungry, but they start shaking and they start sweating, is that because something's wrong with their hypothalamus?
No, it's probably that their pancreas is a little more vigorous than it needs to be in terms of insulin production. Because like blood sugar is not part of this system. So blood sugar, that part happens at the duodenum and the pancreas. Mm-hmm. I don't know why some people get hypoglycemic.
Because they're not hungry.
No, they're not hungry. And in some ways, you're right. You can add that to this system and it would help. Shouldn't you get hungry before your blood sugar drops? Mm-hmm. But it's those blood sugar drops, it's not because there isn't enough glucose around. It's that there's too much insulin. It's being whisked away too fast. Does that kind of make sense?
Yeah.
Yeah. So a lot of research going on these two things, but just know there's a satiety center, feeding center, both are in the hypothalamus. All right. Oh, there is also, so this is short-term regulation. This is day-to-day. What about over the year? Well, adipose tissue has a hormone that it releases. So when there gets to be a lot of adipose tissue, this hormone leptin levels go up, and that turns down the feeding center over many weeks. So you eat less and less and less as you get fatter and fatter and fatter. That's how it's supposed to work anyway. In the human, that system doesn't work as well. Like a squirrel, they get fatter and fatter and fatter, as winter approaches, because then they're going to sleep for 3 months. So in that system, it controls their diet. But we're not sure, why people get obese, frankly. But that leptin system doesn't work as well as it probably should. All right, so thermoregulation is another aspect of metabolism. 60% of the energy released from the food that we eat becomes heat. And heat, our body has to be in a temperature range. Otherwise, things start to break down, things start to not work. So if we need to maintain our heat in a particular range, then we need some system for doing that. And that system is thermoregulation. It's one of the things that makes us different than like a snake or a lizard is we can maintain our own body temperature through these mechanisms. So ultimately, we have heat to get rid of. The only time we don't is when the air temperature is much, much cooler than our body temperature is, like middle of winter, so to speak. The rest of the time, we have heat in excess. We have more heat than we need. So we have multiple mechanisms for getting rid of heat. One is radiation. So just like the sun, projects heat over distance, we do the same thing. All objects that are warmer than their surroundings radiate heat. So we have radiation, we have convection. As air moves across our skin, it takes our body heat away. Evaporation, when water goes from liquid to gas, that takes energy away, it takes heat with it as it evaporates. So the human takes advantage of this evaporation on the skin surface when we sweat. When we exercise, we sweat. Why do we do that? You put water on the skin, that water evaporates, and as it does so, it takes heat away with it. So it helps to keep us cool. It's like it's almost like a swamp cooler for the body. And then conduction. Very small amount is lost in this way, but this is direct contact. So, your feet on the floor, your body heat is going into the floor as long as the floor is cooler than you are. So we've always got heat to get rid of. Why is it important? Because outside of our normal temperature range, things start to become deranged. In other words, bad for you. So, here's the normal temperature range, 98.6 degrees between 36 and 37. Everything's normal. As we go up from that and down from that, we get increasing physiologic effects. So even a change of, 20 degrees from 98 to 118, you're dead. Same in reverse, you're dead again. So we have to thermoregulate in order to keep us in our very narrow temperature range. So first thing in the morning, you have what's called your basal body temperature. That's the lowest temperature that you have anytime throughout the day. And it results from being inactive for all that time. Skeletal muscle generates heat. So when you're sleeping, you're not generating as much heat as you are when you're up moving around. So your temperature falls just a little bit. All right. And I think we'll stop there for today. I will email everybody about what we're going to do on Wednesday because I may cancel class on Wednesday because I don't think we're going to need all that time for review. Okay, so let me look at the review and see how long I need, and then I'll let you know about Wednesday, okay?